So, thank you. First, I would like to thank the organizers of this IBRO event for the invitation. Let me introduce myself. Saber Masmoudi. I am professor on human molecular genetics in the Center of Biotechnology of SPACS, and I am the head of the laboratory of molecular and cellular screening processes. The laboratory is working in the field of health biotechnology. So in relation with neurological disorders, we have several collaboration with different clinical departments, in particular with the Neurological Institute of Tunis, but we are also members of a seed project, is an European capacity building project that aims to improve the molecular diagnosis of encephalopathy, in particular using next generation sequencing. So for this presentation, I am accompanied by Miss Amal Souissi. Amal has already 5 years of experience with next generation sequencing. Because of the pandemic situation, we have an online workshop, and the organizers proposed a question response format. We are also informed that you are a heterogeneous group with different backgrounds. It's not so easy, but we will make this workshop cool and helpful. So now we will start. So we prepared 2 types of question to test prerequisites, but also to assess and evaluate the assimilation of the presentation. So we will ask Carthaginics to launch the first series of questions, please. So 2 minutes. For the first series, we have 3 questions. First, how big is the human genome? Second, what is the estimated size of the human protein coding exome? And third, how long did the Human Genome Project take? It's not that participants are not able to participate to the question, no? No.
Sorry, we can't vote.
You can vote?
No.
Okay.
I
will ask you, excuse me, could you present yourself?
So moderators and can't participate, but the participants can.
Okay.
So, Peter Consulting, is it okay?
Yes, only participants are allowed to do. No.
Okay. Okay. The result, please. Okay. So for the first question, 72% for 3 billion, 76 for the Exxon, and finally 18 years, 60%. Okay. So for the first question, the 70% of 72%, 3 billion pays. Yes, yes, it's okay. You noted? Okay. So as I see, the majority. Okay. So for 70%, effectively the human genome contains approximately 3 billion base pairs, which reside in the 23 pair of chromosomes, and each chromosome contains hundreds to thousands of genes. But I believe that for the exome we have not
Yes.
the good response for the. In fact, for the exome, only approximately 1.5%, so only 3 million base pair of the human genome encode, which encoding 20,000 to 20,000 to 25,000 protein coding gene, which is called exome. So 3 billion base pair, and for exome is around 3 million base pair. For the third question, it's not 18 as the majority of you has, but it takes only 13 years. So this human genome project, which is publicly funded with around $3 billion, was initiated in 1990 and completed in 2003. In 2001, a draft version of the Human Genome Project was delivered. And as you see, 13 years, because it was a long way, the method used was BAC-based Sanger sequencing, which included several steps. So DNA is fragmented into pieces around 2,000 base pair. Then these pieces of DNA fragment were cloned in bacteria to prepare the large quantity of DNA. Then back were cloned to be mapped to the human genome. Then the back library, which covered the human genome, were cloned in plasmid with a fragment length around 2,000 base pair, and then sequenced using Sanger sequencing. So it's a long way that took already 13 years to have, for the first time, the first complete idea of the Human Genome Project. This introduction made only to show you why at this time we see that other technology for sequencing human genome are needed. So now we go to the second series of questions. They are sample and they are related to the different generations of sequencing technology. So please, the second series of questions. So it's now it's the same. The second, please. This is the first series. Thank you. So the second series is sample, and it contains 4 questions. First, how many versions are present for the first generation of sequencing? Only Sanger sequencing or many types? Only 2 sequencing generations are currently available? Yes or no? Are currently available sequencing generations require amplification by PCR? And the last, second and third generation sequencing are also called next generation sequencing, high throughput sequencing, massive parallel sequencing, or the responses are correct. So 2 minutes also. Yeah. So, okay. So for the first question, only 66, 66% said that there is only single sequencing. For the second question, yes, for 28%, only 2 generations are currently available. Now, so you have a response of this question in the last question. Huh? So you need to be more careful. Huh? So in the last question, we asked that there is second and third generation sequencing. Huh? So your attention, please. Huh? Okay, no problem. You noted the response? Okay. So for the first generation of sequencing, excuse me, every time I need to, why? Okay. Okay, so concerning the first question, there is more types of version of first generation sequencing technology. So certainly the Sanger sequencing developed in 1975. In the same time, there is another technology developed by Maxim Gilbert, but this technology was not later developed. So only Sanger sequencing was developed. So as you know, Sanger sequencing is based on chain termination methods. using labeled DNTP during amplification. Then we have electrophoresis to read the sequence of the fragment, and one fragment by electrophoresis. So this is one limit of the Sanger sequencing. There is another technology already used from the first generation, which is called pyrosequencing, described in 1993, and is based on the detection or the production of light signal by a transformation of pyrophosphate. So during the polymerization, there is a release of pyrophosphate, and this pyrophosphate we are able to transform to the light signal, and we can later determine the sequence of a fragment. So for the second question, there is more than two-generation sequencing, and it's due to the progress in different technologies, certainly including molecular biology, nucleotide biochemistry, polymerase engineering, but also to the implication of other technology as surface chemistry, microscopy, nanotechnology, that different technology, in particular second generation and third generation of sequencing, were developed. So this different technology, the first and the second generation of sequencing, as we will show you later, require amplification. But this step of amplification is not necessary for the third generation sequencing. And with the third generation sequencing, and this is the advantage of this technology, that we are able to sequence the original molecule, the single molecule of DNA or RNA. Another major difference between first, second, and the third generation sequencing, that first and second are a short read sequencing technology, but with the third generation, we are able to have a long read higher than 10 kilobase, from 10 kilobase to 2 million pair base. And this is an advantage. First, because these rank reads are useful in particular to detect some specific variation of the human genome, in particular the copy number variation and the large DNA variants. But this technology also allows us to avoid amplification, in particular when we have a GC-rich region, amplification is usually difficult. So briefly, the second generation sequencing. The second generation sequencing, it translates into sequencing multiple DNA fragments. So we are able to sequence in parallel different fragments for different samples, different fragments for different samples at the same time. It also offers greater discovery power, so as we can go in deep with sequencing. So we can sequence the same fragment 1,000 million times. The second generation method can be grouped into three major categorized by hybridization, by ligation, and by synthesis. By hybridization, developed by Affymetrix, is based on array DNA nucleotide of known sequence. So we use array DNA with a known sequence, and we hybridized with labeled DNA fragment to be sequenced. And by hybridization, we are able to determine whether the labeled fragment matches the sequence of the DNA or not. It's almost the same with delegation. So for delegation technology, it involves hybridization and ligation. So it's more specific than this technology, which is based only on hybridization. So hybridization of labeled probe and anchor sequencing. The labeled probe encode 1 or 2 known bases. So each time we arrive with a specific probe with one specific nucleotide, in particular for BGI, or 2 nucleotides for EBI. And then we anchor, and if there is a ligation, we will detect the signal. But this technology are practically abandoned, except for the BGI, which is based on hybridization. And today's technology are based on sequencing by synthesis. And sequencing by synthesis, there is 2 approaches. First, the cyclic reversible terminator. It's the same than Sanger sequencing. So it's based on the terminator molecule in which ribose group is blocked. So that's preventing elongation. So DNA template is primed by a sequence, as for Sanger sequencing. This is the prime. Okay? Then polymerase will add the nucleotide, the labeled nucleotide, labeled but also blocked. And the 4 nucleotides labeled with different colors are added in the same time. If there is an incorporation, so the surface is imaged with high resolution microscopy, and the signal is detected. And according to the color, as for Sanger sequencing, the base is determined, and so on. Okay. Just after the fluorophore is detected, the blocking group can be removed, and a second cycle will be initiated. This is the technology used by Illumina and QIAGEN. Another approach, also sequencing by synthesis, what we called single nucleotide addition, is based on a specific signal produced during polymerization. As we showed previously, there is the pyrophosphate group, and the pyrophosphate group liberation can be transferred to light signal. There is another technology, we will discuss this technology later, based on the detection of the hydrogen group also released during the polymerization. So for this technology, the 4 nucleotide must be added iteratively. So for each position, we have 4 cycles. First, we add, for example, adenine, then guanine, then cytosine, then thymine. But this technology does not require the NTB to be blocked, and to be labeled. The most inconvenient of this technology is inaccuracy, in particular where there is repeated nucleotide. This technology was the first developed for NGS by 454 company and Roche, but in 2013, this technology was shut down. So now we move to the third generation of sequencing. We will go back later to the second generation with more details. The third generation of sequencing, the first commercialized technology was developed by Pacific Bioscience, is also referred as single molecule real-time sequencing that, as I said, enables long fragments to be sequenced. The platform used dye-labeled nucleotide and flow cell. So massive parallel sequencing is also possible with this third generation sequencing. So we can multiplex samples, and in everywhere we have a polymerase attached at the bottom of the microwell, and during polymerization, as we have labeled nucleotide, each time a nucleotide is added, we have also a high resolution imagery system that detect the fluorescence and determine the sequence. Currently, there is 3 sequencer commercialized by Pacific Bioscience, the sickle system, and the sickle 2 system. The major difference between both systems is the number of reads. So for the sickle system, we are able to read 1 million fragments at the same time. For the sickle 2 system, we are able to read around 8 million fragments at the same time. So 1 million, 8 million. The output for sickle system is around 20 gigabase. Remember that, for example, the human genome is around 3 gigabase. The sickle system able to produce around 20 gigabase. So if we use the sickle system to sequence the human genome, we are able to sequence the human genome, and we are able to read this genome only 6 times. But with the sickle 2 system, it's 8 times more. There is 8 times more reads than the sickle system. So Pacific View is already used for different applications. Whole genome sequencing. For human genome sequencing, the sickle 2 system is used, but these systems are also used for RNA sequencing and for targeted sequencing. Targeted sequencing is a sequencing of specific region of the genome. For example, the exome, we are able to sequence the exome of the human genome. This technology is also used to study epigenetic modification. The second technology used for third generation sequencing is developed by nanopore. Yeah, this is. So unlike the big machine of PacView, the first developed system of nanopore is the Manion, and this Manion has the size of an iPod. The principle of this technology is based on the fact that each nucleotide has different size and electrical properties. So we don't use polymerase, we don't use biology, so technology is based on electrical properties of the different nucleotides. So during sequencing, and according to the size and the electrical properties of the different nucleotides, different signals are produced when the single strand DNA base by base is going through what we call the nanopore. Currently, there is also 3 systems. The Manion, the Gridanion, and Prometanion. So if we look for a Gridanion system, so as I said, the system has the size of an iPod, and with this system, we are able to sequence and to produce up to 42 gigabase by one analysis. Okay, so the read is can reach around 4 megabase. One read, 4 megabase, and the output is around 42 gigabase. But so for the Gridanion is the same system. Gridanion is the same system, allowing to analyze in parallel 5 flow cells, and the Prometanion is the same system, allowing to analyze and to sequence in parallel 48 flow cells. So we sometimes also, I will ask Amal, sometimes it's confusing. So we are sometimes found in the publication the 4th generation sequencing, yeah?
Yes, so for the nanopore-based DNA sequencing, it's also sometimes considered as the 4th generation sequencing. Otherwise, other publications also consider this in situ sequencing as a 4th generation. So in some publication, the 4th generation sequencing talks about the in situ sequencing, and in some other publications, the nanopore-based sequencing is also called the fourth generation sequencing.
Okay, thank you, Amal. So we come back to the question. I hope now it's more clear. This is the question related to the fact that second and third generation sequencing are also called next generation sequencing, high throughput sequencing, massive parallel sequencing, and all answers are correct. So when we use next generation sequencing, it's related to second, but also to the third generation, and all this technology are high throughput sequencing and allow to sequence in parallel several fragments, thousands of fragments, and several samples at the same time. So, to finish, as definition, next generation sequencing collectively describes sequencing approach that have reduced the time and cost and tremendously increased the sequence output when compared with single sequencing. And as shown here, just to have an idea about the, excuse me, how? Okay, so just this and this, thank you. So just to have an idea, this is published on the National Human Genome Research Institute in the NIH. So you see this is the cost per megabase of DNA sequence before the introduction of next generation sequencing between 2006 and 2007. So it cost $10 million by megabase. Today, 10 centime by megabase. This is per human genome. So $100 million with Sanger sequencing, and today sequencing one human genome is around $1,000. So now we will go more in deep with the second generation sequencing. So please be careful, because the next series of questions will be asked after the introduction and the details related to the second generation sequencing. So I will start first with the technology currently commercialized. So first, the technology of Thermo Fisher Scientific, which is based, as shown on cyclic, which is based on synthesis. So during the polymerization, there is a release of hydrogen, and using high sensitive sensor, the production of hydrogen can be measured. So as shown with biosequencing, nucleotides are not labeled and are added iteratively, A, then G, then C, and then T. So today, currently, there is 3 sequencer commercialized by Thermo Fisher. So ion gene CBS5 systems. So we have 3 systems, and the high throughput, the maximum throughput for the different system is different. So the maximum throughput from 15 to 50 gigabits. One advantage of this system, that a range of chips with a number of read from 2 to 3 millions to 103 million read could be used with the same system. So with the same system, we can use for a targeted sequencing, for sequencing a small genome, or to sequence, for example, human genome. So the same system using different chips, yeah, can be used, and the maximum throughput is variable according to the system, but also according to the chips used for sequencing. So there is also the PGM system is a DX system for in vitro diagnostics. So 3 systems for research and 1 system commercialized for in vitro diagnostics. So this is for the first system. There is 2 other systems. The generator commercialized by QIAGEN, but it's a closed system. We have now is a hybrid system using technology for clonal amplification of used by Thermo Fisher, but for sequencing the system used Illumina technology. We have its closed system. We have not a lot of idea about the system, and we need to use reactive specific for the system. 3 also sequencers commercialized by BGI in China. So the MGI-6 50, 400, and the high throughput for genome, the MGI-6 T7. So just to have an idea about this system, for example, so for this system, the size of read is around 100 per base, 100 per base, so it's a short read. We are able to perform single and sequencing or paired and sequencing. So we are able to sequence in one direction or in both direction. In the system, we are able to introduce 2 flow cells, and in each flow cells, we have 10 million reads or 500 million reads, and the output is from 10 to 150 gigabits. Now I will present the leader in the market. The leader in the market for the second generation sequencing, the short read next generation sequencing, is Illumina, and Illumina commercialize different types of sequencer with different throughputs. So for benchtop sequencer, so benchtop with the maximum output from 1.2 gigabase to 120 gigabase, maximum read length around 150 per base, that can reach 300 per base for the MySeq system. It's important to note that for the MySeq system, we have three versions. We have a version for research only, but also we have a DX system for in vitro diagnostic, and there is also FGX system currently commercialized by Virogen, not by Illumina, which is used for forensic analysis. So MySeq with system, MySeq DX system, and also MySeq FGX system for three different applications: research, in vitro diagnostic, and forensic. Excuse me. Oh, sorry. So for Illumina, there is also a large scale sequencer for application as large whole genome sequencing, plant, animals, and the high throughput. For example, for the NovaSeq system, Bayram is 6,000 gigabits. So we have 20 billions reads at the same time. So now we will come back to the just a precision. I said that the Illumina is the market leader for sequencer, and one advantage of Illumina that several companies is an open platform, and several companies are producing solutions for Illumina sequencing, in particular for the library preparation. In fact, for second generation sequencing experiment, there is at least 3 steps. First, the library preparation, then the clonal amplification and sequencing, and finally, data analysis. And Amal will details the different steps of second next generation sequencing.
So thank you, Professor Saber. So after having an overview about the sequencing generations, we're going now to focus on the second generation sequencing. So as said by Professor Saber, so the overview of the second generation sequencing that starts with the library preparation and then followed by the clonal amplification and sequencing and then the data analysis. So starting with the library preparation, which is a critical step and it's really crucial for successful sequencing. So what is the libraries? Actually, libraries are collections of short DNA fragments which are completed either by fragmenting this DNA or by multiplex PCR and have had adapter sequences attached. So as seen in this picture here, so this is our DNA insert that will be sequenced later on, and these are the adapter ligated from both ends. So as several factors may influence the quality of libraries, such the DNA input amount and quality, as well as the GC rich content. So it's really important before every next generation sequencing before the manipulation, the quality and quantity assessment is really important for the DNA input. So this is why we use different methods. Here we can see the spectrophotometry using the nanodrop. So this technology, this technique would provide the purity reports directly supplied here and also may allow us to the identification of specific contaminants such as the RNA, proteins, or phenol and salts. So this is the first step that we do for the DNA input, so to check the quality. After that, which is the second technique, which is the flyometry, it's really recommended in each next generation sequencing protocol, and this is because of its specificity, that selective measurement for double-stranded or single-stranded DNA or RNA and proteins. So this technology is very sensitive and measures up to some picograms per microliter. So these both techniques here are used for the quality and quantity assessment of the DNA input. Otherwise, also during the library preparation here, some points of checking the quality and quantity during the manipulation are also needed. So in this part, we are using the capillary electrophoresis, which is the bioanalyzer. It's actually an automated electrophoresis which provide a high resolution separation. So we can use both techniques like the flyometry and the capillary electrophoresis for the checking point of our library during the library preparation. So in the DNA library, we talked about the both adapters ligated in both ends, and this is, as I said, the DNA insert. But also we can find either a single read sequencing primer for single end reads, or also we can find two read primers. So this is in orange, and this one is in blue for paired end sequencing. Actually, for Illumina sequencing, we can use this paired end reads, so the fragment is read in both sides. So we can see this is why we are using this two primer sequencing. In other technologies, they only use one sequencing primer, so they only read their inserts from one side. So this is the difference between the single end read and the paired end reads. So the final aim of the library preparation is to obtain our nucleic acid fragments with adapters attached in both ends, and also with this read sequencing that are essential for the sequencing step. Okay, so as I said, for the library preparation, the whole DNA couldn't be sequenced directly like this. So we should either amplify the fragments by PCR amplification. Then we are using using region of interest specific primers with overhangs adapters that they are going to amplify our regions of interest, and at the same time they are going to insert these adapters in both ends for the sequencing. So another way to prepare our library is to fragment our input DNA, and at the same time also, as we said, for the adapter ligation. So different techniques are used for the DNA fragmentation. We can either use the mechanical fragmentation or also use the enzymatic fragmentation. For the enzymatic fragmentation, also different techniques are developed. For example, the Illumina technologies have developed a specific enzyme, which is the transposon, that is able to do this segmentation, which is at the same time fragmenting this DNA and inserting the adapters at the same time, and this is called the segmentation step. In other technologies, a simple digestion with the restriction enzymes are used, and then a DNA ligase is needed for the ligation of the adapters. So here we can see this is before the segmentation. We have the whole genomic DNA, and here after the segmentation step, we have fragments of DNA with adapters in both ends. So we talked about our inserts, we talked about the adapters here from both ends, we talked about the specific read sequencing primers, and now we are going to talk about the indexes. So the indexes are specific sequences which are going to be inserted with the adapters by a specific PCR. So with this specific PCR, we are going to insert this index primer with the adapters here. This P5 and P7 are actually specific for the Illumina technology, and we are going to detail this after. So here the index sequences, as I said, which are also called barcodes, which are specific for each sample. They are going to be fixed to each sample, and this will allow us to pool all the samples together and then facilitating the sequencing of different samples at the same time. So after the pooling and once sequencing, here we are going to have different output data, and these indexes were going to be used for the demultiplexing step after the sequencing, and thus we are going to find each sample with its aligned by its side. So here we are going to use these indexes in order to for multiplexing different samples at the same time.
Okay, thank you, Amal. Please, the third, now we come back to the questions. So please, the third. Please, the third. Okay. So the third series of questions include how many questions? 1, 2, 5. Okay. Okay.
Okay, so going back to our questions, so we have here 5 questions for this section. Some of them are already explained, and the others will go back to the explanation after the answers. So if we can see the results, thank you. Can we please see the results if possible? Okay,
Thank you.
So which of the following choices refer to the top 2 companies? So the majority have answered that Illumina and Thermo Fisher Scientific. Yes. Which of the following statements would be important for preparing next generation sequencing libraries? And the majority also have answered that all of the above are important. Yes. And for the library preparation, PCR amplification could be performed for the DNA input, or both enzymatic and mechanical fragmentation of the DNA input are possible, or DNA fragmentation is a key preparatory step for DNA sequencing, all of the above. For the indexes, so they allow many libraries to be put together. Yes.
Yes.
And which of the following steps would occur on Illumina flow cell surface? Bridge amplification, sequencing by synthesis, or cluster generation, or even all of the above? And this is the correct answer, and then we can detail the other things. So moving on to the answers. So that's okay. The Illumina, Thermo Fisher Scientific are the most refers to the top 2 companies. The following statements, all of them are needed and important for next generation sequencing library preparation. And for the library preparation here, also the PCR amplification could be performed, and enzymatic fragmentation, and so on. And for this one, the indexes, which are called the barcodes in next generation sequencing, so they allow many libraries to be pulled together. Here, just quick definitions for every single technical word, maybe. Let's see. So next generation sequencing libraries are the collection of similarly sized DNA fragments with adapters in both 5' and 3' ends. And for the next generation, adapters are complementary to the short sequences present on the surface of Illumina flow cells. And for the next generation sequencing, primers, which are adjacent to the adapter sequence, indicating the starting point of the sequencing grid. So also this is for the following steps. All of the following steps will occur in Illumina flow cell: the bridge amplification, the sequencing by synthesis, and the cluster generation. So going back here to this next step for our generation sequencing experiment for the clonal amplification and the sequencing. This is the MySeq, so we are going to focus on the Illumina technology. So this is the MySeq Illumina, this is the MySeq flow cell here, and this is the incorporation buffer. Here we have our pooled library, so this is the pooled denaturated library already prepared. And it will be loaded here in a MySeq reagent cartridge. So what is the flow cell actually? So the flow cell here is a thick glass, which is with adapters here. This is the sequence which is complementary to the adapter already ligated in our library. So the cluster generation will occur on the flow cell here. So the first step, this is our denaturated pool here, which is going to be fixed to each adapter, because here there is a complementary with the adapter already ligated, and these are the sequences on the flow cell. Just to note, this single molecule will bind to the flow cell in a random pattern, so there is no rules for this, so this is just random. And as I previously said, so here this is the fixed library, and then this is the formation of the PCR bridge, and this is here the cluster generation. So the sequencing, as previously explained by Professor Saber in the first part, so will be held with the sequencing by synthesis using the chain terminator reversible, the chain reversible terminator. So each time this nucleotide are labeled and blocked, so with once it's fixed, it will be imaged here, and then the fluorophore and the block will be removed away, and the cycle will begin for the second nucleotide.
Sorry, Amal, probably we will skip the video.
Okay.
We'll skip the video to go to the next step.
Okay. So once sequencing is completed, what is next? So now let's move to the dry lab, and this is the data analysis lab.
So different files are produced after sequencing. First, BCL and FASTQ files with the same information, the sequence and the quality of sequence. But the presentation of the data is different between BCL and the FASTQ. And this file will be produced after demultiplexing the different sample according to the index, and after trimming the step to eliminate all the adaptator and to retain only the insert sequence. Two other files are produced during alignment of the sequence obtained with a reference sequence, and finally a variant files with the different variant according to the comparison with the reference sequence. So getting representation, we explained the output of the sequencer. So as we mentioned before, the output will depend on the size of the selected read. So here, for example, we selected 150, here 250. So the output for the same MySeq sequencer will be different. The output also can vary if we use single read or paired read. The output of the sequencer will also depend of the number of reads on the flow cells. For example, with MySeq, you are able to use V2 version with 50 million reads, or V3 with 25, and the output is different. And from MySeq, for example, to HiSeq or NovaSeq, the output is also different. So here we rapidly details some metrics used to design the success of our sequencing. So first, the Q-score. So the Q-score or Fred quality score is the most used common metric, and is logarithmically related to the Pascal error probabilities. An example, usually researchers used Q30. So Q30 is equivalent to the probability of an incorrect base card 1 in 1,000 times. That means that the probability of correct base card is 90.9%. So, as I said, the Q30 is usually used because starting from Q30, the error it will be around 0.1%. So this is considered, as I said, a benchmark for quality in next generation sequencing. Low Q-score can increase false positive variant card, which can result in inaccurate conclusion and higher cost later for validation experiment, in particular using single sequencing. This is a FASTQ file format. As I said, we have sequence and we have quality indicated by letters, symbols, and each letters and symbols, for each letter and symbols, we have a specific Q score. It's also important to define the percentage of on-target sequence. So if we sequence exome, we will see the percentage of the sequence which correspond to the on-target sequences. This is very important, and this is condition our selection of the size of the red during sequencing. So, for example, if we performed fragmentation and we have a fragment around 150 per base, so we need to sequence with at least 100 per base, and then our target base would be theoretically 100%. So Q score, then the percentage of on-target sequence, then the coverage that include the breadth of the coverage, but also the depth of the coverage. So the breadth of the coverage is the percentage of bases of a reference genome that are covered with a certain depth. So if we said, for example, that we performed exome sequencing and we didn't detect mutation, you have to ask if your sequencing covered all the exome, or you have they have to define the percentage of exome covered during sequencing. So covering is important, but the number of times the sequence or a base is covered is very important. Typically 20 to 30x are usually used for diploid organisms, but it will depend if you are searching for recessive mutations, or you are searching for dominant mutations, or you are searching for somatic mutations. So a good method used for estimating the required depth of coverage is to begin with 20x and divided by the expected allele frequency. So if you have a recessive mutation, homozygous recessive mutation, so 20x is sufficient. If you have a dominant mutation, so 50%, so you multiply 40, in this time 40x is important. If you are searching for a somatic mutation, for example, with only 5%, so you need already 400x coverage depth. So another important element is the uniformity of coverage. This uniformity, it depends in particular on the GC person. For example, here we have two exons of the same gene with different percentage of exon, and the depth of coverage is different from one exon for exon 2 and for exon 1 and exon 2. So rapidly we will, it's okay, yeah? So the fourth question, please. The first series, please. Okay, thank you. Amal.
So for the fourth series of questions, we are going to ask about the next generation sequencing output and metrics. So the next generation output forms, which forms are they? So what is the maximum output size for one run? It depends on what exactly, on sequencing platform or number of reads or read length or all of the above. And what are the next generation sequencing performance metrics, which are already explained now? And what is the definition of the coverage? So if the answers are ready, so please show us the answers. Good. So for the next generation sequencing output, so the first one is the FASTA-Q, the second is the BAM files after the alignment, and then after the variant calling, we have the BCF. And for the maximum output size, it really depends on everything. So in the sequencing platform, and also on the number of reads, and also in the read length. So for the metrics, we have the on-target percentage, we have the coverage, depth, and the uniformity of coverage, as well as the Q scores. So all answers are correct. For the coverage also, so all these answers are correct. So this is it's the average number of sequenced base that align or cover known reference bases, and also it represents the number of times read maps to genomic target, and also determines whether variant discovery can be made with certain degree of confidence and particular base position. So
Okay, we
so we can skip the answers, so that's fine. So okay, after sequencing we have the output, and then but it's also we also have some challenges for the data interpretation. So if we can have the last series of questions, please, before explaining some points. Thank you. So as I said, so how many output variants are expected to be identified after sequencing a whole human genome? Is it 4 million DNA variants per individual, or target region sequencing, or whole clinical exome sequencing? Or all the answers are correct. And according to the American College of Molecular Genomics guidelines, how many classes are possible for a variant classification? Is it 2, 3, 4, or 5 classes? So once the answers are ready, you can see the answers if possible. Okay, so thank you. So how many output variants, either 4 million or 1 million, we are going to see. To reduce this huge number, so all possibilities can be carried out. And according to the ICMG guidelines, we are going to see also how many classes. So if you, your answers are different, so we can see here now some explanations. Okay. So how many output? This is really a huge number of variants that we can find after sequencing the whole human genome, which is around 4 million DNA variants per individual per reference. So there is a summary of single human genome. So actually, it's really some big numbers of single nucleotide variants, which is like 3.5 million. As an example, there are some premature stop variants, 80 variants for the stop loss, 10 non-synonymous, 11,000, yes, 11,000. Some splice site, index, frame shift, and in-frame are possible in a single human genome sequencing. So to reduce the huge number, actually, we can carry out all these possibilities. So whether we start or we base our filtration with a good filtration strategy, or we can also not sequence, we are not also, I mean, we can also start sequencing some target region only. We are not, we don't need every time to sequence the whole genome, so we can focus on some regions of interest that we are interested in, depending on the disease that we are working on or any other specificity for the work. We can also specifically sequencing either the whole exome sequencing or also clinical exome sequencing by focusing on only the exome regions, either the whole exons or maybe also like for example, the TrueSight one, which is focusing on 4,000, more than 4,000 most clinically relevant genes. So all these possibilities we can carry out in order to reduce the huge number of sequencing. So this is some techniques for the target enrichment. So either we can use the alloplex, which is enrichment by fragment circularization. We are going to focus on some region of interest only by circularization of the DNA, or also we can use the enrichment by PCR amplification using some specific primers. So we are going only to enrich or only to amplify the sequence or the specific sequence that we are going to be interested in on the sequencing step. Or also we can use some probes that will allows us to enrich by hybridization capture. So we are going to capture the sequence of interest using this using a specific probes. Okay, so according to the ACMG, there is 5 classes. Actually, this after finding a huge number of variants, so this American College of Medical Genetics and Genomics and the Association for Molecular Pathologies have developed some standards and guidelines for the interpretation of sequence variants in order to facilitate the understanding of this pathogenicity of the variants. So we have here 5 classes, either the benign, likely benign, or variants which are called VUS, which are with this uncertain significance, or likely pathogenic, and finally the pathogenic classes. So this recommendation here we can have are classified according to some types of evidence related to the population data, to the co-segregation, to the competition and predictive data, the functional data, or any other information about this variant. And then it's classified according to the strength of this criteria. Either it's supported, either it's strong, supporting, moderate, or very strong. So once we are here using this criteria to interpret and to understand the pathogenicity of our variants, we can classify the variants in one of these classes. So here also we can use this versome, which is using also the ACMG classification to make it easier. So the versome here displays automated variant classification according to the guidelines of the American College of Medical Genetics. And this is an example. For example, we can put our variants in this versome, and then we have all this table, and all these criteria are then explained. So here, for example, for this variant, it has this criteria PM1, PM2, which are moderate, and two supporting criteria for the pathogenicity, three other supporting, the PP2, the PP3, and the PP5. So here, the explanation, this is the automated criteria. Let's take an example. For example, here this PM1 refers here to the present, to the, sorry. Yes, this criteria informs us that this variant is in a hotspot region that contains many missense and many pathogenic variants. For this one here, it refers to the frequency of this variant in the GNOMAD database, or any other information about the pathogenicity of this variant that might help classifying variants.
Okay. Thank you. So